Actually, this session will be recorded and broadcasted, as you can see here. So by coming here, you're all agreeing for this. And this session is really about discussing about how to do and how can we coordinate this activity. So we really expect you to speak out and share your ideas.
We also have virtual participants, and feel free to type in. There are people who monitor your response.
Can you move to the next slide, please? Yes, this is the QR code that you can use to just connect yourself to the Google Doc page. And actually, if you just go to the agenda and if you click to the link, then you can still go to the Google Doc that we have an initial idea about what to discuss about in this session. So please take your time for doing that.
And then please also feel free to type in your comments into the Google Doc so we can all see together.
Okay, now can we have our main Google Doc on, please?
So as we wrote in this talk, we will discuss about autoimmune disease and infectious disease. It's driven by complex interaction between the host genetics, environmental exposure, tissue-specific immune regulation. So it's a critical cellular processes in this context, in this regulation of immune pathways and inflammatory signaling, cell-cell communication, and also pathogens that interacts with the host cell. So quite complex. And as you can see from the previous session today and yesterday, we've been discussing a lot of autoimmune diseases and also infectious diseases that are important both globally and some regionals and some locally important ones. This breakout session, we will explore the strategies to integrate diverse type of data spanning single-cell spatial multi-omics technology. We try to unravel the mechanisms that drive disease heterogeneity. This session will emphasize how diversity across population, genetic background, and environment shapes immune response to the disease and disease progression. Move up a little bit. These are the key questions that we start thinking about that we will discuss today, that Jong-Eun will introduce this set of questions.
Yes, so I would like to start this session by freely discussing about what kind of autoimmune or infectious disease that we have in mind currently and what can we focus as a consortium. So if you have any idea about your specific disease of interest or your idea about how to coordinate the studies between diseases, for example, we actually have listened to the beautiful talk from Yukinori since this morning about actually the infectious disease and autoimmune disease can be interacting with each other with the genetic factors. So we would love to hear your opinions. And for some of you who are working on some sort of the disease, could you kind of speak up and introduce your main interest? If you raise your hand, then
you can raise your hand and the microphone will go to you. If no one starts, let me start a little bit as a primer. So my particular interest is in tropical infectious disease, particularly dengue infection. And I've been talking to a lot of colleagues here. And dengue infection is very common around the equator line, so tropical areas. So in Thailand, even here in Singapore, but also not only in Asia, but also in Africa and South America. So I've talked to Patricia. She's also said it's a bad problem there, and they also have some cohorts of these viral infected samples, and we might be able to collaborate multi-regional. So I usually work within the Asian community, but this is a general meeting. It's a great opportunity for us to work across region as well. So I think that's one of the projects that we could work together in Asia, South America, and Africa. Yeah. Any idea?
Or anyone who's working on infectious disease, like virus disease, could you raise your hand if you are working on like actual infectious disease samples? Waradon, I know that. Mm. Yeah. Please.
Hi, good afternoon. I'm Jared De La Rosa from the Philippines. For us, it's actually pulmonary tuberculosis. So we're actually establishing the first single-cell core facility in the Philippines, and our focus is actually on post-TB bronchiectasis. So we're trying to recruit cohorts with the entire spectrum of pulmonary tuberculosis, those that were cured, and then those that unfortunately had post-TB sequelae, such as bronchiectasis. So yeah. I'm not sure if there will be other people here or interested in PTB.
That's great. Our collaborator in Thailand also have TB. Are you working on systemic or pulmonary? Pulmonary, do you have like tissue cells or do you blood?
Yeah, we're working with blood.
I see. Great. So, dengue, TB.
Yeah, I know there's quite a lot to discuss, but I worked on COVID, nasal airways, and now our atlas has half of the data set is COVID-19. So interested in mechanistics of COVID-19 and how it compares to other viral infections as well. So both in epithelial tissues and PBMCs.
That's great. COVID-19 is pandemic, so everybody got affected, and we think different population responds to the same virus very differently. So even though we have already quite a bit of COVID-19 data, I think it's still important to integrate globally and how different populations react to the same virus differently.
Yeah, so potentially, like we could organize the infectious disease-oriented like bio-network, and we have many different kind of the infectious agent. Like some people can focus on bacteria, some people can focus on viruses, and some other focuses on parasites, which obviously exert clearly different paths of the immune reaction in our body. But then we still don't know about like how do they compare with each other, and why do our immune system like respond differently to different kind of pathogens. So I think that would be a really good opportunity for HCA to like coordinate and then make a like global infectious disease sort of atlases.
So besides infectious disease, anyone work with vaccine response or anyone work with autoimmune disease? Would you like to share?
So we've been working in autoimmune disease, common autoimmune disease, rheumatoid arthritis, colitis, and looking at if we can find common programs among autoimmune diseases that occur together, so things like rheumatoid arthritis and lupus, or diseases that respond to the same treatments like psoriasis and psoriatic arthritis. So looking for kind of common programs in blood or tissue in autoimmune disease.
Yes, please.
Yeah, I'm Hideki Ueno from Kyoto University, and we do many studies on influenza vaccines in a different time course. And we take a system biology type approach. We use a multiple mix like proteomics, metabolomics, single-cell sequencing, T-cell sequencing, bulk T-cell seq, and yeah, we do many stuff.
That's great. So common autoimmune disease, rheumatoid arthritis, SLE, psoriasis, and also influenza vaccination response. That's great.
Is there anyone who's interested at like genetic diversity and how it affects the prevalence of specific autoimmune disease? Because for different type of the autoimmune disease, there are different type of the genetic sort of preferences. So yeah.
Okay, maybe the geographic and genetic diversity. I'm focusing on multiple sclerosis. Multiple sclerosis it's a kind of a disease named for the wealthy white men, not familiar, right? So they are much higher prevalence in the high north area of the, but still not clear why they're so diverse in incidence. And genetics, my intuition itself do not explain the differences. So there should be something different or environment. So I'm very interested.
So besides genetics, we also have to consider environment and exposure.
Do you want any other ideas?
We can go through,
Yeah.
and we actually have quite a list of questions, and we can always come back if we can think of other diseases.
Can you actually go, like move this further up a little bit so that we can see other questions?
Yeah, and please feel free to type in. If you don't feel like speaking out, you can also type in response to each question. And it would be great if you can put your name and your country, institute, before your comments. Thank you.
Do you actually have a writing authority here? Does anyone try typing here?
Yes, editing.
Yeah, feel free to edit so that we can see that you do have a editing authority.
We're gonna try.
If not, Esther, please, could you modify the authority? Yeah. Okay, so then maybe we can move on to the next kind of topic, which is about how to understand the disease mechanism using the atlasing approaches. So we can think about gathering, like, what kind of omics data that we should gather, and what kind of formats should we make. There can be, for the both infectious disease and autoimmune disease, there are, I think, at least two mechanisms that we need to consider. One is genetic, so genetic diversity between people. The other would be the environment. So we are being exposed to the different antigens, food antigens, or different type of the vaccinations, and then different types of the pathogens. So I think if we try to make an atlas of the infectious and autoimmune disease, the metadata integrity and the depth would be really important. So we kind of want to hear about what kind of omics approaches we should consider and what kind of metadata that we need to collect. So if you have any idea about that, please share your idea with us.
So as a global community, I think we have a great opportunity to have different geographic diversity as well as ancestral diversity. And also certain countries, as Patricia might comment some more, also have diverse ethnic background within the same geography. So for example, maybe in Brazil, you have also Asian population. We could compare that to Asian population who live in Asia and try to tease out the environmental factors as well as the genetic factors. Any idea?
Yeah, so thank you. It's a huge challenge because you really want to capture genetic diversity, and immunity is one of the factors being shaped the most by in human evolution by local selective pressure. So that should be a component for sure that you want to consider. But then on top of that, you have the depth of the immunological profiling that you want to get. And so perhaps thinking about single cell RNA or some proxy of it as a kind of scalable assay that you can use to map differences, genetic differences across populations. And then, of course, deep dives into, deeper profiling, maybe specifically targeted to conditions of interest. And I think perhaps really thinking through local, diseases of local importance or, conditions of local importance. That's where you want to focus work. And the approach we've been thinking also about is, exactly the problem of benchmarking diversity across experiments that are done in different settings, right? If you do experiments in different countries, you'll always have that confounding, that you need to control for. And so how you thinking through experimentally what are the protocols that will allow us to benchmark data across different settings is very important. And perhaps thinking also of, in some cases, building studies where we capture ethnicities in different environments so that you can study the environment, the interaction of genes with the environment. So it's complex, but I think having a scalable, universally applied resource anchored on genetic data and then local design studies would be the way I would think about this. Thank you.
Thank you. Great comments. Maybe we could share a little bit about how IDA works in different countries and how we share all the protocols since sample collection, even the reagent lot number, we try to harmonize all the protocols. And we actually found some differences if we use different, even centrifuge speed. So I think it's a very, very important point on both the data generation and then data analysis pipeline. So at least from our experience in IDA, harmonizing the protocol from sample collection to data generations is very crucial. And another part that we use is to use a single reference that distribute to every country so that it will be incorporated into all the reaction, then we can compare during data analysis. Any other thoughts on how could we improve or, yeah, Gordon, please.
But then when we think about diseases, then we might need to think whether, like PBMC would be the most representative of, like any manifestation of diseases. Should we consider granule size at all? And whether the current protocol would include like cells that we expect them to be. So based on our experience, for example, we found that even like freezing the cell, we lose a lot of plasma cell. And maybe for healthy people, they may be fine, like not a lot of plasma cell to begin with, but then if we think that the diseases are mediated by antibody responses, maybe this is something that we should consider. And then if it's like we need to have them fresh, maybe like logistically is much more difficult, but maybe we need to rely more on like fixation-based protocol. So I think like even thinking about the type of tissues, right? Like, like blood, convenient, but then even with blood, right? Like, like what types of cell would matter, and that would dictate the protocol because it's going to be more targeted, right? Because we need to think about what cell types that would matter in like the context of diseases that we are interested in.
Thank you. Also, when we talk about disease, oh, please. Go ahead.
Yeah, so the other thing I was thinking is you mentioned spatial technologies, and I think that's quite important for mapping the cross-tissue specific immune signature interactions that you're looking at compared to single cell suspension. So thinking here of identifying niches of interest, and if you're looking at ligand receptor interactions or pathogenic cell types, where exactly do they sit in the niche, what kind of neighboring cells you have around there. So leveraging their single cell resolution for spatial technologies, I think will really help. And then you mentioned metadata as part of building immune atlases or other atlases. We spent quite a lot of time trying to come up with what we called tier 2 biological metadata. Actually, this could be relevant across different atlases with the aim to ultimately combine these atlases into creating a whole body reference map. So in there, I think if we think of, let's say, COVID biology, we can come up with fields that are relevant. And then for other, let's say, lung bionetwork or so on, that's also relevant. And this can help when you put the PBMCs and you make cross-tissue atlases for immune responses.
Thank you. So spatial and cross-tissue atlases. I think maybe when we have the first version, of the healthy ones and started to have some optimized protocol that would be very helpful to go beyond healthy and go to the disease. And having working with dengue viral infection, I think inclusion criteria and time points and severity classification are something that are really critical, really important. And unlike healthy PBMCs. So if we were to work together, I think these are also the inclusion, exclusion criteria and the time points and how we classify the heterogeneity of the disease in clinical setting would be really important aspect as well. Do you want to share?
I've got one point to add.
Kevin?
That's great. So I think particularly for autoimmune disease, familial relationship must be a metadata that must be recorded because siblings of people with type 1 diabetes or rheumatoid arthritis are not technically healthy. So I think when it's difficult how to define healthy actually, even considering that. But if we could start collecting data like that, then at least maybe for version 1 of the draft, yes, we group them out all as healthy, but then later you can go back and say, yes, actually, there is sub-definitions of sort of healthy, but not really. Then you can then build upon that. Yeah.
Definitely, even healthy, we need to define what is healthy. This is very important subset. Yeah.
Yeah, this brought to the question of like what is truly healthy and how to actually liaise with the version 1 like healthy immune atlases with and expand that like with a continuous manner with infectious disease and autoimmune disease atlasing. And I think there maybe we need to care a lot about the comorbidities of the different disease and because now the next goal of the human cell atlas is the clinically applied atlas. So we need to really think about how to integrate all the like clinical information, like in-depth clinical information about history of the certain patient or donor, what kind of infections, previous infections has been gone for each donor. Those information might be really, really important. But I'm wondering in current immune network, like metadata scheme, how is that being incorporated? If Gary or Anna, could you share the current thoughts and what could be the next type of the metadata that could be incorporated considering this? So what kind of metadata is currently being incorporated in the healthy immune atlas and to expand it with infectious disease? What do you think we need?
If I may, I'm also involved in the Immune Atlas, so I can give a quick overview. So as Anna-Marie mentioned, we do have quite an extensive tier 2, so that's kind of like the official name for this type of metadata, tier 2 metadata schema. But currently it's targeted, disease-wise, it's targeted more towards COVID. So the healthy immune V1 is not just healthy. So we're also including like half of the cells actually come from COVID patients. And I think that was also done intentionally because we want to kind of provide like a reference that can where at least one disease is present, and that might give us like a better representation even for mapping other diseases. So back to the actual metadata we have. So we have multiple different fields for COVID, so some similar to also the metadata that you mentioned. So some time course, for example, how many days after, let's say, infection or some estimate infection was the sample taken? Did we have multiple samples? If we did, then how far are those samples taken? And then we have a couple of approximations, or as far as we can get, because we can't always get the most best resolution of the information, but some estimation of the disease severity or maximal severity. And I think in that case, yeah, that's pretty much the extent that we're going to. And then of course some other diversity-related factors like sex, age, and I think things that also apply across different organs.
Yep. Thank you for sharing, Gary.
So that for COVID, we have had difficulties because we're collecting data from lots of different centres, and you can only really use data that's already been collected. It's not really realistic to ask a clinician to go back through the notes, especially at atlasing level studies where these are hundreds and hundreds of patients. And because all autoimmune diseases are going to have different measures of severity, the severity of lupus is different to the severity of rheumatoid arthritis. I think one of the things about the metadata is to be pragmatic about what is realistically going to be routinely collected in clinical practice and what we can ask people to collect that is going to be useful. So while we can be hugely ambitious about the metadata we'd like to have, I think it's important to be realistic about what we can actually have.
Thank you. Let's highlight the importance of the metadata. And also, I think if we were, so the genetic diversity network also creates some metadata that recommended for the HCA community as a whole. But I think maybe for particular disease sets such as autoimmune or infectious disease, we perhaps need more field of the metadata that we should fill in that's specific to our disease of interest. And may I ask if anyone also routinely have clinical lab in all the samples and also keeps certain plasma and serum for all of these samples and data, just in case, for example, with history, we never know with the history of previous infection or vaccination for certainty. So having these extra tubes of samples might be quite helpful. Just want to hear from the audience.
Thanks, Ponpan. I think I have an answer followed by a question to that question. We run diagnostic labs across the country, across Bangladesh. And yes, for some of these outbreaks or other endemic diseases also, we do collect very, very detailed metadata. And we have a huge biobank of maybe 100,000 samples, from pathogens to biological samples, depending on what we are interested in, and for many diseases, both plasma, serum, whole blood, and maybe about 200 to 300 variables that we collect for each of those samples. Having said that, now, when we started this biobank about 15 years ago, having said that, now, when we started doing single-cell genomics, we realized how crucial it is to carefully collect samples, carefully freeze the samples, ensure every cell inside is preserved. We go to the community with a box of dry ice, whereas when we are collecting samples in hospitals, basically a sample comes from the hospital ward to the lab, stays on the bench for all the diagnostic tests for a few hours, and at some point when someone has time, takes out a little bit of sample, puts it in -80. Yes, we have a biobank, but can they be used for single-cell analysis that's going to yield good enough data, is my question.
Yeah, please.
Hi. I work on the Heart Atlas and the Organoid Atlas, and I think metadata-wise, yes, the most difficulties we have is that basically people have to rewrite their metadata, I feel. I mean, either they don't have it, then they won't have it available, but I mean, one big thing is getting someone to sit down in tier 2 case with medical background knowledge, which is also not every lab, and basically just write this all down from the information they have. So what I would suggest is that we need some kind of, we need to send out the tier 2 lists, maybe updated for each organ very early to each collaborator, and everyone who's involved in the HCA should just write down in that schema, because then when we ask for it, if we ever include the data in the atlas, and then it's quite easy, I think, because you're in a clinical study, you're writing this down either way, but what's annoying for people is having their Excel table and putting it in our list, and that takes ages every time.
Yeah, I'd like to follow up on the sample handling and the technical effects question as well. So coming from the integration team and that kind of being the main type of technical variation that we are trying to deal with or correct out, I would say one thing we learned that was very crucial is also collecting metadata, not just for clinical relevance or biologically relevant information, but also for all of the technical preparation, like sample prep, library prep. Mainly, I think sample handling could have quite a big effect, and I think that's something, unfortunately, we don't always have full insight to in our current setup, just due to whatever has been published or whatever information is still available. So I think that just highlights also the importance of collecting these technical metadata, and that's also what in HCA we define as tier 1, and that is something that should usually be applicable across all organs. And I wanted to add to that as well that the methodological aspect is also quite important, right? There are multiple methods out there that do try to attempt removing batch effects in different ways. And I think the technology is there, but from my point of view, the main challenge is how do we know that these methods are doing a good job, or how do we know that these methods are doing a good job for the specific question that we are trying to ask? Because in the end, we always have to deal with a trade-off of good batch removal, but also like loss of biology. And then also going back to the disease, that also highlights the importance of considering not just cell type variation, but also the disease variation when we're actually building these atlases. So that we just have a bit of an estimate of if we're correcting out a disease effect or not, and if that kind of gives us a good representation of the disease in our atlas. Or also in cases where we map diseases to the atlas, that is also something we will want to quantify.
So I also believe that problems that we had with the HCA data that were mentioned now are not going to happen when we're talking about the diseased ones, because they're going to be drafted specifically for the kind of disease that we want to work with. So the metadata is going to be selected by experts and not by molecular biologists or technical people who were interested in the technology in the past as it started, right? So I think it's going to be different. I don't think we're going to have this kind of problems. And the idea is like every time we have a disease that there is going to be, like somebody mentioned, from the beginning, what kind of metadata is like tier 1 and 2 and 3 for that specific kind of disease. So I believe it's going to be well done. I'm very hopeful for that. I don't think it's going to be the same problem that we had now. And also everybody now is like more conscious about the ancestry diversity, the diversity that is necessary, and like they're also going to keep that in mind, which is also something that was not done in the beginning because that was not the point, right? You didn't have enough data sets to worry about that, but now it's something that it's in everybody's mind. So I really think that it's not going to be a problem. I think that the biggest issue is always going to be what disease, and how to get people to agree on doing something focused, like something that is relatable for a larger group of people, so.
I think all those were very important comments, and just practically to advance, I think we should design this rather prospectively rather than retrospectively, because as we see, there are some sample handling issue and there are some unexpected metadata that we might need to collect. So usually, I think IDA was quite good example that people just tried with the kind of first flagship project, like pioneering project, to just trying to collect PBMC in a uniform manner, and then trying to kind of come up, deal with all the technical effect. We, for example, surprisingly found that although we have used all the same kit and experimental kit, but what's happening in the hospitals actually might matter, because we realized that, for example, all the samples collected from the Korean hospital was slightly tilted in terms of the cell proportions. And we believe that might be due to the centrifugation or something. Very difficult to figure it out. So I think we should try, and for trying that, maybe we can coordinate by the sort of disease types. For first, virus people gather together, and then parasite and bacteria, and then autoimmune disease. And then here, people start to make a network on their own and just build up the first flagship like pioneering project. And then also lies with the Gary or Anna or all the other healthy immune atlas people to connect their newly collecting prospective data with the existing atlases to enrich them and build it further. And with that regard, could you actually scroll this a little bit up to see other comments? Okay, we are so far from the computer over there. So just to raise another interesting and also very difficult question is, all these infectious disease and autoimmune disease is heavily linked with the antigen specificity and also our immune repertoire. And this is still a mystery how to solve the immune repertoire and antigen like specificity problem. Even the AI is not solving that with the structure on the protein structure. So I'm curious, like what kind of ideas that like you guys have on how can we deal with this, and what kind of data should we collect to solve this problem if we have a great opportunity to profile all the infectious and autoimmune disease across the globe? So that's another question that we had. Any ideas on that? Is there anyone who's interested in making like immune repertoire atlas? Who's doing? Yeah, please. Yeah.
Microphone, please.
Yeah, please go on. Yeah.
Okay, so I'm not working specifically on antigen immune repertoire, but just from our experience from building the immune cell atlas, I think that even on the single cell level, we have some signal of certain genes. So we actually noticed that, and we noticed that kind of hindered our like annotation effort. But I think there could still be some scope in when you do feature selection or just with the types of assays we already have. And for some of the studies that we're using, some of them also publish TCR, BCR assays. So I think in terms of data availability, of course, it's always better to collect more, but I think that there's quite a lot that you could already use and then maybe try to put into context of multi-study atlas yes, and I believe that's also like what the V2 Immune project is kind of looking at as well. So yes, and I think what might be interesting to explore is just to see even on just a readout, like single cell readout, if we can already see some signal. And seems like we could, but we haven't followed up specifically.
Thank you. And Kevin?
Yes, I have lots of ideas. So for context, I write software to analyze immune repertoire analysis. One of the things we discussed before is like clonal expansion and like how at the single cell level, whether that clonal expansion, are we actually picking up all the clonal expansions or are the singletons, so the single BCRs and TCRs, are they just low level sort of sequencing depth? So, but they're actually expanded clones. And I think there hasn't actually been a robust sort of matching of bulk to single cell repertoire. But there's something maybe with enough cells we can actually do. The other thing is on the other comment about data availability. Surprisingly, there's not a lot of B cell receptor sequencing single cell level data, even for healthy bone marrow, where there's several human bone marrow atlases out there right now, but none of them report their BCR sequencing data. So I think that's something that is a low-hanging fruit that should definitely be curated and published, because then that will lead to other things that could lead to other things and so on. And yeah, okay, I'll say that for now.
BCR.
I was just going to add very briefly as well, one of the things that we don't routinely necessarily do is HLA typing of donors, but for interpretation, we should start doing that routinely in donors for interpretation. Because I think we would hope in autoimmunity that if we had sufficient numbers and we could match to HLA background, we would begin to find some shared motifs. We haven't done it so far, but I guess that would be an ambition.
Yes, actually there was written, I think, below that we should also collect really accurate HLA genotyping for all those patients. Although using single-cell data, we could impute at some level the HLA types, but I think we should really think about having the genome coordinator here, because we need to really think about the genomic diversity and not only about HLA types, but maybe also just TCR, BCR locus itself, and then many other GWAS-associated areas. So, Yuki, do you have any idea about how to coordinate that part? In Japan, you have collected a lot of genome transcriptome paired data set, right? So,
I think most of the case, before starting the samples, we should definitely need the protocol, right? Shared protocol and eligible protocol for especially PBMC or split time or something. And genetics is very easy, but even RNA extraction strongly depends on how we. So this is very important. So to predefine the recommendation, especially we, for example, discuss something granulocyte, right? If you want to focus on granulocyte, each type of. So I think one of the most, because we each other collected so many samples, but they are different, difficult to integrate. So this is a miserable situation. I think it's predefined or some making the recommendation, okay, how we should prepare for pandemic, how we should prepare for the means, and for eligible cross-country collaboration. I think maybe one of the practical very important step.
So maybe from another angle, so even before talking about disease, right? Even with the HCA atlas now with a healthy one, I'm always curious. We have PBMC data, but then we have T cell, B cell, but then what are they reactive to, especially the memory T cell or the memory B cells, right? Are they against commonly infected viruses, CMV, EBV, herpes simplex virus, I don't know, hepatitis B, or influenza, get vaccine, get infected every year. So I guess having some kind of baseline of the lymphocytes, reactivity of lymphocytes, would be another step, not even before talking about the disease, right? Because we know that in autoimmunity or infection, this T cell might kind of interact, having prior immune responses and then having contribution to, I don't know, severity of autoimmunity. So maybe thinking about healthy, but then, I don't know, a panel of some kind of antigen-specific detection in some way, so we can detect some of the commonly known antigens, or even autoantigens, I don't know. But then starting with the disease to have some baseline idea would be great.
CMVs and EBVs, we are kind of relate that with sometimes autoimmunity, but still we don't know with the statistical power, how does that contribute to the onset of each disease. So I think that's a very good idea. And maybe also we could talk about, we don't necessarily have to only think about single cells and single cell transcriptomics, but with the blood, there are many things that we can actually measure, even from serum. We can measure some antibody levels and existence of some cytokines. There are many blood proteomics that are being done with the, for example, UK Biobank. So I think we can also think about what can be collected from the samples if we do, this kind of a study.
It's also about autoantibodies being all around, even in healthy people, and for certain autoantibodies could contribute to severity of certain infection and maybe contribution to some autoimmunity as well.
So to make this really variable, I think it's good to have all of them listed. Maybe we can do the survey of what can be recorded, and then we just circulate it all around the community so that we have the same things in mind so that we can run the flagship project before the next meeting so that we have some ideas about what can be collected. So.
Hi, so maybe this is more about a question or comment that I have. So I think it's not really talked too much about the treatment paradigm. So I think for autoimmune immune patients of different subsets, or maybe patients coming in as naive patients that have not been treated. So should there be some kind of focus where we, because of our atlasing, to look at the initial stages so that we try to stay away from the confounding effects from the treatments themselves, because you can imagine the first line, second line might make things very complicated. Yeah, so wondering if there's any focus, maybe we start with naive patient if it's possible, and then if there's money, and then we can follow with longitudinal studies and see how this changes, responders, non-responders, so on and so forth.
Yeah, I think that's very important point, like, because these diseases are not only heterogeneous, but it's very dynamic. So we need to think about those dynamics and the sampling points. And for example, the SCATE story that we just heard about, I know that there's a study which they have sampled the naive before 3 month samples and then did a drug response prediction. And then this naive situation, status is distinguishing the responders and non-responders. So we really need to think about longitudinal sampling and considering incorporating all the 3 month information to the metadata so that we can have a good power in terms of the analysis.
Yeah, thank you.
Any other comments? Any bioinformatic difficulties can you foresee about these kind of integrations? Or I'm also curious about how to apply the spatial omics on this disease, because I see a lot of cases of the spatial omics applied to the cancer biology or developmental biology, but not yet many of them apply to the infectious disease or like inflammatory disease. So if anyone has a kind of plan to do that, please share with us.
Hi, so we have projects on the cystic fibrosis. Like we do the probe-based VISIM for like lung cystic fibrosis, and actually the capture rate is a bit low. It's a bit challenge to like get the bacteria in the lung samples. So my next plan is also do like lymphatics tuberculosis. So is this plan for doing kind of 3D spatial transcriptomic models to looking for host pathogen interaction? So I'm not really sure for now, but just want to share with it.
We have some planned projects on lupus nephritis. So I have actually a poster on it outside, but we are planning on doing spatial transcriptomics on patients who have lupus nephritis. And one of the difficulties we have is because you sample kidney biopsies and they're needle biopsies. And if you're lucky, then you pick up a non-fibrotic region, but if it's extremely fibrotic, then the cellularity is quite low. So the gene capture is low, you don't end up getting a lot of information. So the one challenge is staging the disease, because you want it to be early so you can get early disease dynamics. The second challenge is getting a clean biopsy section so we can like actually sequence it. The third is to make sure that we get all sections of the kidney, because when you get a needle biopsy, if you're lucky, you get the glomeruli. If you're not lucky, you just get the tubular interstitium. So it is useful, but I think the glomerular inflammation and scarring is also something pathologists use to stage the progression of the disease. So I think those challenges are there. But we have some plans to get treatment agnostic lupus nephritis patients. So it's difficult to find them, but that's on the roadmap for us.
Usually the problem with this kind of disease is lack of samples for the spatial omics, and the needle biopsy is really difficult actually to have a comprehensive understanding of the tissue. So that's one problem, and maybe we can think of the other utilizing other model organisms for the dynamic spatial profiling, and then also we can think of a bioinformatic like task to kind of link it with the human disease status. So,
to add on to that, so I work in pediatrics where autoimmune diseases is rare, right? You might get that one patient that has that rare disease. So then there's no way that I could create an atlas of that disease with that just one person. But then, anyway, so I was gonna ask, what do people think about privacy then? Because if that disease is so rare, their repertoire, their gene signatures could potentially be identifiable if the human cell atlas or the disease atlas publishes it. So maybe there needs to be some safeguards for those type, especially for pediatrics, where that's going to be the situation, I think.
Yeah, actually, so I think to give like one suggestion what I guess could be done also, or a little bit like how we envision one use case of these types of atlases is that ideally, having like a huge cohort of cells that we do collect, and maybe having some level of representation of potentially also a couple of autoimmune diseases. So I think there's definitely more scope for that. One idea would be for a user, or like as a use case, is that if you have like your own data, you should be able to use the atlas to kind of augment your data and actually put your, let's say if you only have one sample or like very few samples, put those samples into context with the wealth that we have in the atlas. And even if it doesn't fit 100%, but I think to some degree, you might be able to recover more signal than if you were just to look at your sample by itself. So I think that could be just one practical solution with the tools and the methods and resources we have at the moment. And I think in that case, you also don't necessarily need to, so it depends on, I think you'll have full control over like your own sample, whether you, and you wouldn't necessarily, that wouldn't necessarily require building like a de novo atlas with very few samples, but you can kind of leverage existing atlases. Just wanted to mention,
Yeah, if there's no more comment, we are about time to finish. But I think we kind of missed one very important point that we also wanted to discuss, which is about microbiology. So all those pathogen side, like metagenomics, what kind of omics approach should we apply and how to collect those information could be very important. And actually inviting microbiologists to this HCA consortium might be also very necessary. And also all the infectious related immunologists who has actually immune experts on like interpreting the data. So with that, I would, let Ponpan to finish.
With host and pathogen interaction, that would be really another layers of complexity, but I think it's still it's an open area that's exciting to work on. So with that, I think we need to start wrapping up, and thanks everyone to be here. We did discuss about the disease of interest spanning from infectious disease of local importance such as dengue, TB, vaccine response such as influenza, and pandemic COVID-19 response, and also various autoimmune diseases, the common ones such as RASLE, psoriasis. When we talked about, as a consortium, we also discussed a lot about metadata, the importance of metadata, and how we harmonize the protocol, and how do we scale up without too much technical difficulties, and how do we apply this to the community and the hospital setting in order to minimize the technical variation.
I think that was most of it wrapping up our discussions. And I think actually Ponpan is really a right person to discuss if you have any kind of plan on making an atlas study because of our experience with the IDA studies. So, so let's gather together on this topic from now on. And thank you all for joining the discussions.
Thank you.